All right, I think we're going to get started. So thank you, everyone. We're really excited to have you all here today. Looking forward to a great set of COVID-19 research lightning talks from our members of the COVID Information Commons community who are joining us here today. This is a program that is brought to you by the Big Data Innovation Hubs as part of NSF's Convergence Accelerator program, intended to bring together a research community that is working on solving the many different aspects of the pandemic. And we have a wide-ranging group here with us today. So if I go back to my slides here. We're going to start with a brief intro from Florence Hudson, who's the Executive Director of the Northeast Big Data Innovation Hub and has been the project lead on the COVID Information Commons, giving you a little bit more context about the program. Florence?
Excellent. Thank you, Katie, for running all of these COVID Info Commons webinars. You're doing a great job. And welcome everybody to the webinar today. The COVID Information Commons started in May when we received a COVID Rapid Award from NSF. And the Northeast Big Data Hub, where Katie and I work along with our students who are on with us today, worked with the other 3 Big Data Innovation Hubs from around the country to create this COVID Info Commons. We did the original launch in July with 2 search mechanisms, the 2 that you, one of them you see here, which is a Research Explorer machine learning map, as well as an NSF Simple Search customized search by directorate. And then on October 30th, we actually just launched a new search mechanism, which combines the NSF COVID award information with a PI database. And Katie will talk more about that later, but there's a survey for NSF COVID Rapid awardees, PIs, that they can fill in and then add their information. So we have a very rich context around not only your plan for your award when you got it, but actually URLs to results you have, your ORCID ID, all sorts of cool information to enable more collaboration. And this is available to everybody. Anybody on the planet can use it. Actually, one of our PI Lightning talks in October, one of the presenters, Sarah Bowman from the University of Buffalo, was contacted by somebody who actually is in South Korea, whose mentor is in the UK, and she's having a collaborative call with them coming up. So this is available around the world, and please feel free to share it with everybody. You're a very important part of this. Everyone in this community, the researchers, the students, the industry, government, higher ed, everybody. So we're delighted that you're part of this. Share it with your friends and colleagues. And I'll pass it back over to Katie.
Great, thank you so much, Florence. We're excited to talk more about that later in the program today. As just a reminder, we will be first going through and hearing from our 6 different researchers from around the country who will be highlighting their COVID-19 research. We encourage you to share your questions in the chat or by joining us via the Slack link here. We will not be taking questions immediately following each presenter's speech in the interest of time. We will have a dedicated section for Q&A following the last speaker prior to the demo that Florence mentioned. So you can save your questions for then, or you can send questions in the chat, which some of them may be able to answer there or answer live later on. So without further ado, we will get to our speaker lineup today. As you can see here, we have 6 different speakers from around the country who will be highlighting their work funded through NSF as part of the COVID-19 Rapid Award Program. We're going to start off today with Debra Laefer at New York University. So I will stop sharing now and let her jump right in. Debra.
Super, thanks so much. I'm Debra Laefer from School of Engineering at NYU, and my collaborator on this project is Tom Kirshner from the School of Global Public Health. And we got one of the very first rapids, and we sent out a big team of undergraduate and graduate students to medical facilities out in the New York area. So what was the goal? The goal was to try to understand at a really hyper level what were people doing, things that you couldn't pick up from closed circuit television. You couldn't pick up from just looking at people's cell phones. Things like, what was the gender of this person? What did they touch? Were they wearing PPE? So we had 16 human observers, and we were out there at the peak of the spring outbreak. So we had our first observer on the ground on March 23rd and the last one on May 19th. As you can imagine, it was a pretty stressful, crazy time. So our students got in time when they could. They were each assigned to a specific hospital or urgent care in an area in which they could walk to. We tried to select students based on the fact that there was nobody else working in their area. So you can see on the right-hand side, we had 3 facilities up in the Bronx, a cluster of them in Manhattan, here in Brooklyn, and then here out in Queens. So we tried to get a kind of a good distribution. The students were told to go out anytime they could, up to 20 hours a week. Ultimately, they collected over 5,000 records of over 6,000 individuals. So what were they doing? They're standing on street corners in front of mostly ER entrances or the main urgent care entrances. And you can see here, there's a food cart, there's lots of things to touch. So this is where the student was standing. Here we're out at Elmhurst. I should say this is Mount Sinai in Lower Manhattan, and this is Elmhurst out in Queens, one of the most impacted sites. And the students were asked to take a smartphone and to basically randomly select somebody as they came out of one of these facilities, to stay across the street from them, not to talk to them, not to touch them, not to photograph them, not to video, and to just note what they were and who, where they were going. So what did that look like? So here we have a record. They came out of this area. They used a phone. It has the time that they did certain things. They entered a certain facility here. They then, they note they were wearing a mask. They interacted with a passerby's dog, and they tried to enter the park, but the gates were closed. Here's another one. Female with a mask and gloves came out, entered the Walgreens, came back out. Students were asked to try to monitor the person for up to 20 minutes, up to a mile in distance, or until they couldn't see them anymore. Many of them, if they went into a small store, they felt they were going to come right back out. And so we have that return record as well. This was the data set. As you can see, it was fairly uneven. In one case, we had a facility that was closed. So a student moved over to this facility. So we've kind of doubled the amount. They're color coded by borough, blue for Brooklyn. Here we've got green for Manhattan, and then purple for Queens, and yellow for the Bronx. This data set was released publicly in September. And is available both as coded in a CSV file, individual GIS files, and a grouped shape file for about 20% of that data. We're about to upload another 20% of that data into a single shape file as well. And we can start to see some quite fascinating things. So here, this was, we're looking at mask usage. So about 48% of the records note whether they're wearing PPE, and we can see that women wore masks more than men. And then here, when the governor implemented the mask mandate, we see both greater compliance, but particularly of the men, we really see a much more significant PPE usage. We can see some other things longitudinally. Here, this is just at one facility. Over the time, increasingly, people just stopped touching things in the built environment. Here in black, we see the COVID hospitalization rate. And even as it stopped to drop, people started to become more and more cautious in general, particularly about touching things in the environment. That's this yellow line. But what we notice is in terms of touching themselves, touching other people, that's the line in blue, and touching personal objects, it's much less clear. A lot of times these people are obviously not thinking about these things as outside themselves. So they're coming out of these high-risk medical facilities where COVID is just rampant. And the first thing they're doing, about 15% of them, is taking their phone out of their purse, out of their pocket, and sticking it against their face. By having it all ultimately into a GIS system, we can start to look at some hotspot activity, both in terms of individual routes and some larger zones. So I just wanted to say thank you for this opportunity to present some of these preliminary results. Thanks so much.
Thank you so much, Deborah. I really appreciate that overview. It's definitely on my mind anytime I go out and about in the wider world these days. So appreciate having the data here. Niema Moshiri of UC San Diego will be up next, presenting on his research. So Niema, we're ready to go when you are.
Cool, thank you. So hopefully people see my slides. So yeah, I'm Niema Moshiri. I'm at UC San Diego. I'm in the Computer Science and Engineering department. And I'll be talking about a recent development that we've made called viral MSA. Basically a standard phylogenetics workflow. If you've ever seen an evolutionary history of virus sequences, you start with these initial sequences at the beginning that don't necessarily line up very nicely. And the first step is you do multiple sequence alignment to get them to line up. Lining them up like this tells us stuff about, what is the sequence homology? What are the relationships between the sequences? Using this alignment, you then estimate a phylogeny, and then you root the phylogeny to determine what is the common ancestor. And then there's a bunch of other downstream analyses that you might be interested in doing, things like what are the clusters of outbreaks that are happening, what demographics do we think are more at risk of getting infected by some disease? Yeah, a lot of stuff that you can do. So in general, my focus is on these two steps, the multiple sequence alignment and the phylogenetic inference, which are really the computational bottlenecks. And in this talk, I'll be focusing just on this single step, multiple sequence alignment. So the multiple sequence alignment problem is NP complete, which TLDR, that means we don't have a polynomial time solution. And as a result, many heuristics have been developed to approximate solutions. They're pretty accurate, reasonably. Some of the tools that people might be familiar with are Muscle, Clustal Omega, MAFT. These are some of the common tools that implement them. However, even these heuristics generally scale quadratically with respect to the number of sequences. So in the case of SARS-CoV-2, we've had an exponential growth of sequencing happening where, this is a great thing for us. We're getting more and more sequence data, but the downside is we have to analyze those sequence data and our tools just don't scale properly. So right now, this is actually outdated. This was from, I think, a week or two ago. Now we're almost at 200,000 sequences. So the tools just do not scale to enable real-time analysis. So what if instead we knew in advance, so a priori, we knew that our sequences are going to be super similar, and we already have some high confidence representative reference genome against which we can compare them to. So here I'm showing the reference genome as a green line on the top, and each of those other colored sequences below it are the sequences that I want to align with each other. Instead of aligning them amongst themselves, what I could do is just align the first sequence against the reference genome, align the second sequence against the reference genome, keep aligning each of the sequences independently directly against the reference genome, and then using the positions of the reference genome as anchors, I can merge the individual pairwise alignments into one multiple sequence alignment. So take the first column of my multiple sequence alignment, see, oh, in the first sequence, this is the position that matched, then this position matched in the second, in the third, and then just collapse them together. I can do this for each position of the reference genome, and then now I have a multiple sequence alignment. And the nice thing is that this parallelizes super nicely. Every single sequence can be aligned against the reference independently and simultaneously. And it scales linearly with the number of sequences rather than quadratically. But let me take a step back real quick and think about this approach. My input is a reference genome and a bunch of sequences that are very similar to that reference genome. And my output is an alignment of every sequence against the reference genome. So if people are at all familiar with long read sequencing, this is exactly the same computational problem as mapping long reads to a reference genome. So my question was, can I leverage existing well-implemented read mapping tools to enable this type of scalable reference-guided multiple sequence alignment? So I developed a tool called viral MSA, which wraps around existing read mappers in order to perform reference-guided multiple sequence alignment. I wrap around a few of them, but there's one tool specifically, Minimap2, that's kind of the gold standard right now for what I'm doing. So I only recommend using my tool with that specific read mapper, but I wrap around a few of them to demonstrate that I can evolve this tool naturally as read mapping technologies evolve as well. So basically, you simply provide viral MSA or reference genome and the sequences to align, and then it'll handle indexing the reference genome and doing any pre-processing it needs to do. And it'll call the read mapper and then merge the results into a multiple sequence alignment. And here you can see a comparison of this approach with existing best practice tools. So my tool is the blue line on the bottom, and then the two other lines are other tools. And you see that in general, it's multiple orders of magnitude faster than what people are generally doing right now. And it scales very nicely. And the sequence alignments that we get are very accurate. So in conclusion, this tool that I've developed enables rapid multiple sequence alignment of viral genomes. It's open source, you can get it online. And hopefully, if you do any viral analyses, consider using it. So some acknowledgements: Heng Li developed Minimap2, which is kind of the essence of the speed. And this work was supported by NSF and Google. And yeah, I'll leave questions for the chat or later in the session. I think you're still muted.
Thank you. Thank you very much for sharing that with us, Niema, and for making that tool available. We'll make sure that's shared on the COVID Info Commons website as well, so that it's accessible by the community. So very exciting. So next up, we have Branden Johnson of the Decision Science Research Institute, who is going to talk to us today about perceptions and behavior of Americans regarding the coronavirus. So Branden, whenever you're ready.
Yes. So first, I'm acknowledging my co-PI, Marcus Mayorga, and we're doing a longitudinal panel study of Americans' views. We took a longitudinal panel study is where you're asking the same group of people questions at several times. And we used an online panel to recruit people. You can see we've done 4 waves so far. We started much earlier than most longitudinal studies, February 28th, which was the day that the CDC announced the first confirmed case of non-travel transmission. And we're at this point expecting to go much longer than most, if not all, longitudinal studies, just because we're doing this at much longer intervals, roughly 2 months apart, rather than the more common month apart or sometimes weekly collection. Like the University of Albany study discussed last month by Sam Penta, we're also using the protective action decision model as a foundation because it includes people's perceptions of other actors such as government, not just their own perceptions. But we're building in some other factors, including some that were used in my earlier longitudinal studies of Americans' reactions to Ebola and Zika, which of course on the mainland were much smaller and shorter outbreaks and were in a decline. This is the model that we included in our original proposal. We didn't cover all the potential relationships because this figure was already getting quite busy. But on the right, you can see the threat, action, and stakeholder perceptions that are part of the PADM with regard to personal protective behavior decisions. We also wanted to explore support for various government policies like mask wearing mandates and the like. And then we have a number of upstream variables that you can see there. And as we've been developing these ways, we will occasionally switch in some other measures of other variables that we think will be interesting. But one of the unique aspects of this study is that most of our variables are asked every single wave. So that, for example, we're not just asking, are you following the news about COVID-19? Which information sources, such as newspapers or TV or social media, are you using? But also, which outlets within those media sources are you using? So we can have, for these and the other items listed there, very fine-tuned assessments of whether and how things are changing over time. Some preliminary findings. We found that a model very close to the one that you saw in the previous slide adequately fit wave 1 data on personal protective actions, but it did not fit policy support. Whenever we tried to put the policy support measures in, the model fell apart. And we're not sure yet, because we haven't done the analysis, whether this is an outcome of measuring people before, in fact, there were many official policies, except banning travel of foreign citizens and the like. We have done a multi-level modeling of waves 1 through 3 in terms of threat perceptions. How much do people think they are personally threatened? How much risk is there for the US, for the globe? How much concern is there for local transmission? And we found, contrary to what the Ebola study found, is that there were no individual differences in trends. So there were not factors making some people say have their perceptions of risk go up faster or slower than other people. But we did find a number of factors, including dread and news following, which were also pertinent to Ebola and Zika, pushing people to have higher risk perceptions. And then one of the protective actions we had put in because of some anecdotes and news media stories was avoid Asians as a protective action, even though technically it definitely is not protective. And we found that if we measured people's anti-Chinese prejudice and their degree of tendency for conspiracist thinking and their notions of the efficacy of avoiding Asians and reducing risk, putting those in meant that the original direct relationship between avoid Asian intentions and conservative political ideology disappeared. So finally, I'll just say that I've already received help from Peter Rose, who presented in September on the UCSD big data project, in getting some case data for each of the states and counties from which we've recruited people for our study, so that we will later be able to assess how much that indirect experience might be related to people's perceptions. And we're already talking with some people about trying to get help with content analysis of these media outlets that I show here, so that we can then not only assess the impact of following these outlets, which we were able to do with Zika, but also whether the content of those outlets is perhaps helping shape the risk perceptions we're looking at. So, thank you.
All There we go. Thank you very much, Branden. Really excited to hear that you've been collaborating with Peter, and absolutely encourage folks to reach out and be communicating with the PIs if you're also engaged in similar research. We found a number of these collaborations springing up, which is really exciting. So next up is Professor Ying of the University of Southern Florida, who's going to talk about sterilization of protective masks. So ready to go.
Hello, everyone. Can you guys hear me? Okay, good. So this is Sarah Zhong from University of South Florida. I'm very glad to share our story about fighting coronavirus with corona discharge. So when you hear about corona discharge, it's not as terrifying as a coronavirus. It is actually a very useful tool which we can use to provide a safe and convenient and affordable technology tool for everyone to get safe masks and safe surfaces. I believe every one of us still has the very fresh and the terrible memory of the severe mask and the disinfectants shortage. So this project was initiated at the beginning of this pandemic to overcome this challenge. Even with the time going on, we are very happy to see that the shortage has been released significantly. However, when we monitoring the situation and we interviewed with many hospitals and civilians and self-practice clinics, we find out that almost like 1/3 of them are still reusing their masks. And they are still concerned about the safety of reusing them. And it's true that their concerns are correct, because based on some research data, we can still see a lot of filtration efficiency drop after certain kinds of disinfection treatment. So, but if we look at the microstructure of those masks, we cannot find specific deterioration. The reason for the filtration efficiency drop is actually mostly caused by the loss of the static charges in the masks, which are injected to them during their manufacturing process. And the difference between a mask with static charges and without static charges for a N95 mask, it can be as much as 37% versus the 95%, which it should have. So here comes our solution, is that we are using corona discharge to disinfect and also recharge our masks. So that means after the corona discharge treatment, you can not only kill the bacteria or the virus, you can also restore the static charge it had right after, which is similar as the right after the manufacturing process. So with this way, we expect to see the microstructure no change, but the static charges can be restored and the viruses has been killed. So this is some preliminary data we got. So for the convenience of experiments, we used the E. coli as the mostly used bacteria to test our disinfection efficiency. Up to now, we can reach a log reduction of 6 for E. coli, which means it is 6/9 in the percentage. It is 1,000 times better than the common disinfection technology we are using in our daily life. And also the log reduction on non-conductive surface, which is the most challenging kind of material which we can do disinfection with corona discharge. Now we can reach log reduction of 3. And we believe if we improve our technology further, it can reach much higher log reduction in the future. And also, we tested our effectiveness against the spores, which are the most tolerant, like a bacteria. You can see it's bacteria to disinfect. And so the log reduction now it's reached 3, but in the future, we believe it can be further improved as well. So in terms of recharging, we discovered that even after a treatment of 1 minute or even less than that, we can reach a very stable charging effect. And this stable charge can stay for days and even several weeks. And we also sent out our N95 masks for filtration efficiency test at Nelson Lab. After 15 times of our disinfection treatment, we still can see a filtration efficiency of about 95%, which makes us very exciting to see that, because after that means our treatment can allow masks to be used for up to 10 times at least. We also are working very hard to improve the broader impact of our technology. For instance, we participate in multiple times of social media interviews, and also we made a series of videos to teach the public how to wear masks correctly and how to reuse masks. And also we submitted a NSF STTR proposal collaborating with a local medical device designing company, trying to commercialize our technology by designing at least 2 kinds of devices. One kind of them is to allow us to put our masks in, and after several minutes of treatment, we can take out the mask and reuse them safely. And the other one is a scanning device, which we can use that to scan the surfaces we have contact during our daily lives and reduce the amount of use of disinfectants. But just with this contactless and the scanning technology, we can disinfect our surfaces. And we are trying to reduce the cost of those devices as well. So our target is to make it as less than $50 for the public. So we are still on the way for that, of course. So that's about it, our research. And we are actually very glad this lightning talk has already connected me with Debra, and we're thinking about further collaborations. So I look forward to more collaborations in the future. Thank you, Wen. Thank you.
Thank you so much. And I'm really glad to hear that you guys have connected, and certainly encourage others to be in touch as well. It's really exciting to see this technology moving forward. So next up in our lineup, we have Leila Hedayatifar of the New England Complex Systems Institute, who is going to talk about modeling COVID-19 in the context of optimizing quarantine policy. So Leila, if you're all set, you're good to go.
Okay, hello, can you hear me?
Yes.
Okay, good. Hello, everyone. Here I would like to share some of the results of our work. The goal of this project is to show that the state governments should collaborate with each other, as the states are not necessarily disconnected from each other according to the mobility patterns. And we would like to create an opportunity to optimize lockdown strategies by aligning policies with the individual's mobility patterns. Okay. It's challenging to predict and control the outbreak due to the complexity of human interactions and movements, and also population density, heterogeneity in population density. In the US, we can say that most of the state governments act separately, with most of the measurements and risk definitions being done by administrative patches or borders. Social distancing, contact tracing, and quarantine policies have been the most impactful policies on controlling the diffusion of the coronavirus. There's a critical need to carefully define the borders of areas with different risk levels, considering the location of suspected cases. And we can say that we need to know where the people who were in touch with the infected person were and when. For this purpose, We use mobility data, anonymized mobility data collected with safecraft from cell phones, and created networks in which locations are connected to each other based on the movement of individuals between the locations. We created weekly networks for all around the US. This figure shows the degree distributions of the locations, showing number of the movements to and from each location. And you can see that in the city areas, we have much more number of movements than comparing to the suburban and rural areas. And mobility patterns that we see in that figure can be characterized in three overarching concepts: short distance movements, medium distance movements, or long distance movements that happen for different reasons. A combination of these habits in a self-organized manner form the size and borders of the communities that we want to analyze. We use Luwian method to find or detect the communities in the US at multiple scales. These communities refer to the areas in which people mostly move within the communities rather than the other areas. Here I showed the communities in the first level with the same color. But these communities are multi-scale, it's multi-scale phenomena, meaning that at a finer scale of subdivisions, larger communities divided into smaller communities allow us or policymakers to go into the smaller scales and do the policies and apply the policies in the smaller scales. Here I show the sub-communities inside the communities with that they are separated from each other by black lines in the map. And we also did analysis in the larger scale. We applied the community detection into the networks of the communities and detect clusters of communities that I showed them here with the same color tone. So you can see that we have 5 large clusters of communities that they have more connections to each other than the rest of the communities. The yellow lines here represent the state boundaries. So you can see that, however, in some of the areas, communities are aligned with this administrative borders. In many of the areas, they strongly deviate from the state boundaries and also county boundaries that it shows here with the yellow lines. This shows that why states need to collaborate with each other, because they are not really disconnected from each other. By adding the number of COVID cases on top of the map of the communities, we can quantify the risk of the exposure inside the communities. And this helps us to align the policies better. Here, I zoom into the communities in the previous figure, so you can see that while a large community may have many COVID cases, when we go into the smaller communities, we can see that some of the communities have higher number of cases and some of them have lower number of cases. So those communities that have less cases, they are safer to reopen earlier. And also the other thing that we can consider is that to be careful or cautious about the commutes between the low risk communities to the high risk communities. These are the things that we can consider. And the other things are that by zooming into the communities, we realize some interesting facts about these communities. One of them is isolated communities. We realize that they are some isolated communities, that they are geographically disconnected from the original community, that they are can be universities or vacation spots, like this university that is connected to the community in New York State, or vacation spots that they are disconnected geographically from New York city community, but they are some vacation spot for the people in those areas. And we also have sub-communities within other sub-communities. We realize that there are some areas that people prefer to move inside the community rather than the nearby area. Good examples for those ones are university campuses and also sub-communities in city areas. We realize that there are some sub-communities that have less,
Looks like Leila's frozen a little bit. She was having connectivity problems, I think you said, Katie.
Yeah, I froze up for a moment myself there. Leila, are you there?
Yes.
She's
Yes.
back now. Great.
Okay, great.
Yes. Do you have me?
Yes.
Okay. It's important to study to see that how lockdown strategies and quarantine policies are changing the borders of these communities and these patterns. Here I showed for 6 weeks from February to May, and you can see that at the beginning in February and March, it's a very clear example. You can take a look at the Florida state. At the beginning of the pandemic, it was connected to the northeast cluster in the US, showing that it was a vacation spot for the communities in that area. And then in March, it's getting connected to the community cluster in the rest of the US. And just in April and May, it's getting disconnected from that far distance communities. So we try to study these patterns by more details, but I didn't have time to explain them here. Thanks for listening. I would like to say that you can see much more about our work in endcoronavirus.org mobility maps. And also I would like to say that we have a team of volunteers and are interested in further collaboration. You can reach us in endcoronavirus.org or at nexi.edu Slack. Thank you.
Thank you so much, Leila. Really appreciate that and sharing that information, which we'll make sure that we share with the community as well. So that brings us to our final lightning talk from Xifeng Yang of University of California, Santa Barbara. So ready to go whenever you are.
Can you hear me?
Yes.
Great. Thank you very much for organizing this webinar. It is my great pleasure to report our recent progress. That work is funded by NSF Rapid program. So I'm Xifeng Yang from UCSB. And the goal of this project is going to forecast the daily new cases, hospitalizations, and deaths across the United States for COVID-19 and disease. This work was collaborated with my PhD student Xiaoyong Jing and Professor Yuxiang Wang, both of them from UCSB. As we can see that the number of daily new cases recently increased dramatically. It is concerning us. And furthermore, the number of daily new deaths increased dramatically across the world. It sends us an alert. And our goal is try to do the forecasting of those daily cases and deaths. And we believe this kind of forecasting would be very useful for local decision makers, for hospitals, for governments. There are many factors that are related to the number of COVID-19 cases and deaths. There are so many different factors. For example, demographic factors, population density in a county, in a state, in a city, local business structures, social culture, psychology factors, and the interventional policies. All of those factors have a kind of influences on the contact ratio among people, then on the infection ratio among the people. And these two numbers are going to impact the number of hospitalizations, the number of ICU beds used for COVID-19 patients. And in the end, they are going to influence the number of deaths. And furthermore, all of these numbers are influenced by the disease dynamics. The COVID-19 disease evolve dynamically. And all of these numbers can be represented as time series data, the infection ratio, hospitalization, ICU, deaths. And as we know that time series analysis has been studied for decades, there are many wonderful time series forecasting algorithms. However, in this work, we would like to do something different. We would like to leverage the newest deep learning models and try to build a pure data-driven approach without assuming any kind of epidemic models. So our intuition is like follows. History repeats itself. Like different regions share the COVID-19 and trending patterns, as the spreading rate is usually determined by common factors, such as social interactions, protections, intervention policies. For example, if you want to forecast the situation of the United States, you can refer Germany, France, Italy, and learn a lot. In order to forecast the cases in a certain region, we actually can refer to these other regions where pandemic starts much earlier and just learn from their experience. So this is the intuition behind our model. So let me use one slide to very briefly introduce this intuition. And actually, this is called attention mechanism in deep learning, and nowadays a very successful model in natural language processing, in models those sequence patterns. Suppose we would like to forecast the new cases in California. What we can do is we try to refer the similar situations in other states across the United States, and we regard these states as reference states. And then we can compare the current window in California and compare with historical windows in different states and try to find those historical windows that share the similar trend. And then we can use the follow-up trend to forecast the future cases in California. Of course, you need to do a kind of weighting. So this is where the deep learning takes place. And then we can just do this work again. That's it. And you move the window and check the similarity among this window and weight different similarities, and then using the follow-up trends to forecast the future cases in California. And you can do it for the daily new cases. You can do it for hospitalizations. You can do it for the number of deaths. And it turns out that this simple model, purely built on the data, actually can achieve top performance without using any kind of SIR, SEIR model. As you know that these models are leading epidemic models, which can help us to understand the disease spreading patterns. So it is a little bit surprising to us. We can achieve quite a good forecasting results, at least compared with those models without understanding the underlying disease spreading patterns. So this slide gives you a kind of summarization and results delivered by leading groups across the United States. These groups send those forecasting results to CDC, and the CDC archives those results so that later we can compare the accuracy of those algorithms. And as you can see that our algorithm called ACTS across time series forecasting actually can achieve quite a good performance. These numbers actually measured using WAPE, is the weighted absolute percentage errors. And use the same model, we can do the new cases forecasting, hospitalization forecasting, and death forecasting. And we do the forecasting in the state level. But if there's a need, we actually can do it in the county level if the data is available. So this is quite a surprising result. And currently we are communicating with CDC. Actually next Tuesday, there's a meeting in CDC and we are going to present this result to a group of experts working in this domain. And we are quite new to this area and we are not expert in time series analysis. Our result is available in the following web link. We just submit a paper in archive.org so that it can be shared with researchers much earlier. And we also publish our forecasting results in this website, and the result has been sent to CDC. So this is it for our work, and thank you for your time.
Thank you, Xifeng, for sharing that with us and for sharing the results there, which we'll make sure that we're making available to the community as well. Really appreciate all of our speakers for sharing all of their great insights with us today. I've seen that a lot of the questions in the chat have been answered as we've been going along. Haley, our awesome students, do we have any current questions that have not yet been answered?
Yeah, so thanks, Katie, and thanks to all the presenters for the very interesting research and your respective findings. So far we only have one unanswered question from Sharon to Debra. And she asked, will you publicize or share your data? And also, what's the geographic range of the data?
Yeah, so that data is already available. I can see if I can find the link for it. It's through the Faculty Data Archive at NYU under my last name. And then it covers 18 different zip codes in the New York City area across all of New York's boroughs except Staten Island, because we couldn't find any student researchers there. So, and we are hoping to have further funding on this fairly soon, fingers crossed, and we'll go back to 8 of the 16 main sites and add 3 US sites and 5 international sites.
Thank you so much for sharing that. I'm having a little issue with my share screen at the moment, so I'm just going to pull this up and see if we're able to get that. Are folks able to see this currently?
Yes.
Okay, great. Thank you. So I'm just going to go to full screen here. Okay, so as we mentioned, as part of the presentation today, we are really excited to share a demo of our new feature on the COVID Info Commons website. I'm getting an odd message here. Are you still able to see the shared screen?
Yes, Katie, but if you want, I could do it. Is your laptop
I'm able to see it properly. It does. I get a message that says sharing is paused, but if you're able to see it correctly, then I'm happy to continue.
We are. Maybe you could hit the little easel so that it's bigger. That's the only thing.
Maybe I could, I'm sorry?
Hit the little easel so it goes into the bigger mode, maybe?
Oh, okay. Yeah, that's where the issue is. My apologies. If you would like to share it, maybe that would be simpler because it appears from my view that I'm sharing full screen, but it sounds like that's not coming across, unfortunately.
Yeah, it's okay. Been there, done that.
So while Florence is pulling that up, as we've mentioned, the COVID Info Commons at covidinfocommons.net is the website that we launched earlier this year that shared a number of different resources, including those that you see highlighted here. Thank you so much, Florence. Initially, this launched with access to the Research Explorer ML maps, which were very exciting machine learning powered visualizations, as well as an NSF Directorate specific search. We are really proud that over the past month, thanks to the efforts of the entire COVID Info Commons team, including our great students who are here today, that we are able to launch a new feature that is called the NSF COVID Awards and PI database. And that you see is accessible both at the top link there and in the search bar that is highlighted in the center of that image there. And this database is a great resource because if you want to click to the next slide, it shares information on over 900 different NSF-funded COVID awards and their PIs. That is all publicly available through a very intuitive search interface, making a lot of these details really accessible to you. You can see on the right-hand sidebar, which is highlighted there, that there are a number of different ways you can filter it if you're interested in research coming out of a particular directorate, if you're curious about what's going on at different institutions or in different states or regions, if you're trying to find, something by the PI's name, or you're curious about, what else is my NSF program officer funding or someone else who involved there. There's a lot of different filters, and there's the option as well, once you've pulled together a filtered search, to download the results as a CSV so that you have access to all of this data. So you see on the main page there that there's information provided at a high level about projects, the principal investigator, and their institution. And if you want to click through to the next slide, I'll show you what the award data looks like. So as you can see, you have the information once you click through to the award about the title, all of the different parameters that we talked about on the filter search there, as well as the full abstract and information about the principal investigators who are affiliated with the award. We highlight Peter Rose, who was one of our particular speakers earlier this year in September, as mentioned earlier, because we have his award listed here. And then if you go on to the next slide, we will see what a typical PI profile looks like. So this provides a much richer set of information, both about the PI as well as their research and opportunities for collaboration. And all of this is thanks to the PI survey, which we sent out, the COVID Info Commons sent out earlier this year, and which I know that many of you have filled out as well. Thank you so much. So you see there's information that includes their ORCID ID, the project websites. Many of you have been sharing your project websites during the presentations, and this information here shows where results are available so that everyone is able to quickly access this detail. As well as detailed information on the right-hand side there about the expected research outputs and different collaboration opportunities. I know that there was mention earlier of someone looking for content analysis and so forth. Peter here has, collaboration opportunities that he's looking for, including data and model providers and users and a knowledge graph developer, so that if you're curious about him and his research and you just so happen to fit the bill on one of these fronts, you're able to get in touch in a seamless way rather than having to, know somebody who knows somebody, which is all too often how so much of this works. There's detailed keywords and other data there. I'm going to just include in the chat here a link to the COVID PI survey. As we mentioned, all of this data that is available is thanks to the survey that many of you filled out earlier this spring, earlier this summer, I should say. We were able to build this database with the Columbia University IT team and our amazing student team who really helped in the clutch as we were getting this data loaded into the website. And if you are interested in adding more information about your COVID-19 research on the website, you are welcome to fill out the survey at the bit.ly link, which I have just dropped in the chat. And we will make sure that that is updated and included so that your websites of your results, any papers you want to share, any data sets you want to share, or any different collaboration opportunities that you would like to highlight, all of these can be available to the community directly as well. So we're really excited about this feature, and we're really excited about great things up ahead for the COVID Info Commons community. And it's really thanks to all of you guys as a community who have been so great about sharing your research and getting in touch with each other and making all these great things happen. So thank you for all of your contributions. And I'll just pause here should anyone have any further questions before we wrap things up. I don't see any other questions in the chat, but given the time, I'll keep things short and sweet. Thank you again for joining us all today. Our next event will be held on December 9th at 3 p.m. Eastern, and full details are available at covidinfocommons.net or via our newsletter, which you can access via the website or through the sign up link there. So thank you again, everyone, for joining us today, and looking forward to next time. Have a good rest of your day.